get going then. Most people seem to have logged on now. So welcome everybody, and thank you for dialing in tonight to the next episode of the 2024 State of Play series. I'd just like to thank Derek Stein, who stepped in at quite last minute last session. I was unavailable, unfortunately, but did a fantastic job of moderating the session. So for those of you on Zoom, you'll probably see a notification about the meeting being recorded. Just click OK or agree to continue. And then I'd like to acknowledge an acknowledgement of country. Biogen Australia acknowledges the traditional custodians of country throughout Australia and their connections to land, sea and community. We pay our respects to their elders past, present and future, and extend that respect to all Aboriginal and Torres Strait Islander peoples today. And I'd like to acknowledge the Wiradjuri people on whose land I am and pay my respects to elders past, present and emerging, and extend a welcome to any Aboriginal and Torres Strait Islander people on this call today. So tonight we're going to hear about the different stages about how we get new treatments to patients. We're very fortunate to have two experts presenting tonight who are really quite intimately involved in the clinical trial and drug registration processes. We have Associate Professor Rob Henderson, who's a neurologist from the Royal Brisbane Women's Hospital. He'll talk about MND clinical trials. We have Lucas Tocchini, who's Head of Market Access at Biogen Australia. And he'll take us through the registration process once the drug has had a successful clinical trial outcome. And I'd just like to emphasise that due to regulatory restrictions, Lucas is not able to talk about Biogen products. We'll only talk about the Australian registration process rather than any specific Biogen products that may be moving through the pipeline. And if he's not able to answer a question, Lucas will say that as well. There's some very strict rules around what pharmaceutical company representatives are allowed to say to patients. So as previously, each presenter will talk for about 20 minutes, and we'll have a combined Q&A session at the end. And please submit any questions you have through the chat or the question and answer function in Zoom, or through the comments if you're watching through Facebook. And you don't need to wait to the end to type your questions. Just enter them in as they occur to you, and then we'll collate them all and make sure we ask all the questions to Rob and Lucas at the end. So I think Rob is going to go first. So please take it away, Rob.
Thanks very much, Gethin and Laura. And look, I'm very happy to talk on this topic because, you know, we desperately need new therapies for motor neuron disease. So if you advance next, traditionally, this will be the structure about clinical trials that have been in the past. I'll talk a little bit about the present, and I'll touch a little bit about biomarkers in a clinical trials. But first of all, I'll just give you a little bit of background about clinical trials, as I think there'll be people on the audience who sort of wonder why we haven't moved a bit why we don't move a bit faster in clinical trials. So next. So, I mean, if we knew what the cause of motor neuron disease is, we probably wouldn't have to go right through these phases. And if you knew exactly what caused it, like parachutes, well, you only need an N=1 trial. But we need to go from different phases. And if we go to the next slide, it outlines in a little more detail about the idea of having the preclinical work from basic scientists about why a drug might work in motor neuron disease. And that's called both preclinical. Phase 0 is getting the drug doses right, and that's usually done in a very small study in a single laboratory. Phase 1 studies where patients are involved, but first of all, there's usually healthy volunteers for getting the dose right, and it starts off at a very small dose and checks the drug is safe. And they're usually done in one site. And then many trials in motor neuron disease overlap phase 1 with phase 2. And what you're trying to do there is get some measures of efficacy in the phase 2, as well as the safety in the phase 1. And then again in motor neuron disease, many studies move from overlap between phase 2 and phase 3. And the fact that you've got to do that probably is one of the limiting factors, that you're not going through the phases in a careful way, because people are really trying hard to say, well, if this drug's got some efficacy, let's advance it as quickly as we can. So if you show in the next slide, most MND studies have only ever reached the phase 2 studies. And so that's usually now done in multiple centres, but only a relatively small number, say with 100 to 300. In Australia, we've had a number of phase 2 studies, and there are 5 sites around Australia that collaborate very nicely in these clinical trials. But phase 3 studies where true efficacy can be seen, and they're usually international studies. And then occasionally studies, not so much in motor neuron disease, but in other diseases, phase 4, some studies where you learn whether it's got toxicity or whether it truly works in phase 4. So next slide. So proper phase 2-3 clinical trials are run very carefully, and that's partly because if the drug is successful, then it needs to prove to a regulatory authority that the drug and the way the clinical trial was run stands up to scrutiny. And most clinical trials, by the time they're reaching that, have been published, usually in clearly identified leading journals. So all of them will follow a basic set of criteria, that they'll have adherence to a protocol. So that's like a standard operating procedure. The lingo used is good medical practice. There's a Helsinki Declaration for how people and patients are treated in trials. The study needs ethics approval. These days in Australia, there is a process through the large public hospitals for multi-site approvals, but there is also a relatively quick private approval process through Bellbury. And a key in all studies now is this idea of independent data management control. So the people doing the research are not biased into saying, well, I'm going to I'm going to find a positive result. And they're called contract research organisations. And some of the, the name, one of them that's quite popular at the moment is called Novatech. But they make sure that the researchers are adhering to the study protocol. And that's a really important part of the clinical trial process. So most clinical trials will be clear about the inclusion and exclusion criteria, that they're randomized. So patients are randomized. They don't have to be placebo controlled, and many phase 2 studies aren't. But the people doing the study should be double blind, so not to be influenced by the study result. When analysing the results, there's what's called an intention to treat analysis. So patients are analysed according to when they start the drug as a group, because otherwise that can be an important source of bias, because if patients drop off the study, you're not sure why they dropped off the study. And at the start of the study, you should have very clearly defined primary endpoints plus secondary endpoints, and you're clear about what you're looking at in the pharmacokinetics. And so some studies run a little bit tricky there, because they start off with their primary endpoint, but they actually record what the secondary endpoints are. So next slide. So this all started in terms of any therapy for motor neuron disease back in the mid-90s, with the drug riluzole. And, some of the study methods are fairly similar to how the studies are run now. And they've got limitations, but they divided patients into bulbar onset disease and patients with limb onset disease. And, where they showed in that middle slide that you can see down on the bottom left, you can see that there's a separation between the patients who had bulbar onset disease to the patients who had riluzole and placebo. So that's that middle graph. They're called Kaplan-Meier survival graphs. So on any of those graphs on the far left, they all start off the same. And those in that study, they were looking at survival. And you can see that patients with limb onset disease, it was less clearly a benefit. And usually they'll display the data in different ways. So you'll have a measure of limb function, bulbar function, and muscle strength, and you'll be doing that in both groups to show a benefit. And you can see down the bottom that there's p-values, and traditionally a p-value of 0.05 is regarded as relevant. So next. So there was a second study to look at different dosage, but that has been the last careful look at riluzole. Next. The Cochrane database and some other studies have shown that riluzole probably has a benefit even later in the disease, just as much as early in the disease. But as people on this call will know, the benefit's been modest. Next slide. So a drug I think you're going to hear a little bit more about in the coming weeks to months is Daravoan, which has been approved in the US and has been used quite widely there and in Europe. And there has been 3 studies in total, and there's been a couple of after studies in the US. But the study which was published in the Lancet's probably the main one. And I guess what they did was a study that was restricting the patients who came into the study. So rather than saying anyone with motor neuron disease, they had 137 patients, and they used the earlier study to inform which patients should be coming in. And that some of those ideas have occurred in more recent studies. So patients have a reasonable level of respiratory function. They are declining at a reasonable rate, and they haven't had the disease too long. So, what you can see in the 4th dot point there is that their disease duration was less than 2 years, and they were declining at a certain rate. But that's really sub-selecting patients because, that doesn't apply to probably the majority of patients. And the journal made them put that point in, I presume, that the results don't necessarily translate to a wider population. So it's been around for at least 5 years, and it's more been the intense IV schedule and cost, but it's currently under PBAC consideration. So, I suspect we're going to hear more about that drug, and I can say more in the chat. Next slide. And you can read quite a lot about its mechanism, but it's a free radical scavenger. And so that's quite an intriguing as a possible mechanism for motor neuron disease, so that the idea that there's oxidative stress. Next. So the first of the studies afterwards was dexpramipexole, which was a proper phase 3 study. Next. And you could clearly see how they did the study. You could clearly see who got in, who got into which group, who dropped out. Next. And you could see in the Kaplan-Meier survival curves that there was absolutely no difference between they were on placebo or dexpramipexole. And so in that case, that's looking at the ALSFRS score, which is a 48-point score. There are 12 different domains, as you know, bulbar function, upper limb function, lower limb function, respiratory function. It didn't show any change in that way. They also obviously looked at survival as one of their key endpoints. Next. So, you know, there at the top, you can see that they did look at survival, and at the bottom, you can look and see also they looked at respiratory function. So those remain the key measures for any clinical trial when, you know, and they're very broad, but you would get some information whether there's any efficacy in any way. Next. So that was very disheartening for a large pharmaceutical company to put an enormous amount of effort. And I guess one thing is the cost is huge for the phase 3 studies. They're the order 30+ million to do a proper phase 3 study. So, but they really do underscore that problem that, you know, the phase 2 studies that suggested it might have worked were misleading. But it really did have a major effect on how future studies occurred after that. Next. So I've missed a lot of different trials, but I just want to, and I'll show the next slide as well. The respiratory function is probably the most effective measure that patients with motor neuron disease can have. But once again, it's only one study from 2003 that was a true positive study. So here you can see in this graph all of the patients up in the top Kaplan-Meier curves, and down the bottom on the left are the patients who had non-bulbar disease, and on the patients on the right, patients with severe bulbar disease. There's been later studies that have suggested there still is a benefit for those with bulbar disease if more carefully managed, but the very clear survival, and if you look across the bottom of the bottom left one, in number of days, you can see the survival is practically a year, going from, say, 150 days up to 650 days. So there is a very clear benefit and survival benefit from non-invasive ventilation. Next. So the major issue for the motor neuron disease trials is that, you know, if you try and measure muscle strength, you'll have the fellow up the top who starts off way too strong. And the biomarkers of the disease pathology have got limitations. And it really is a variable disease, as many people who here would know. And only really age and bulbar disease correlate well with reduced survival. And we're recruiting patients to studies when the disease is already advanced. So currently, most trials are using these criteria called the tricals to include patients who are more typically progressing. And they're using those similar things that dexpramipexole looked at, which was how long have you had the disease? Are you progressing? What is your current respiratory function? Next. And, you know, even if we wanted to use genetic markers, which you would think is the purest form of understanding it, what this shows on the right, and you can't see it well, but if you've got the same mutation, same SOD1 mutations, the duration of the disease and the age of death are highly variable for the people who've got the same mutation. So genetic mutations has not been an easy way to try to consider clinical trials. Next. The FDA has been very positive and really has an approach that let's approve therapies for further research. Let's give them a go, but they've got to try to have, they've got to sign up to a proper clinical trial. So that's why you'll read in the paper that there is, an FDA has now approved a new product for motor neuron disease. Next. So the current biomarkers that are used, and I'll talk a little bit about some current trials, are survival's not really as much used as the ALSFRS, the forced vital capacity, measures of muscle strength testing, and then secondary markers which might be things such as imaging markers, trying to assess how many motor units you've got, trying to use blood biomarkers, which are called neurofilaments, is the leading one. And these days, cognitive assessment is poorly assessed in clinical trials, but some trials would say you cannot be enrolled if there is what's called frontotemporal involvement. Next. So a good clinical trial is you've got to recruit earlier. You've got to run the trial for a longer period, 12 months, 2 years. You want to try to have a population that, if you are comparing it with placebo, it's homogeneous, and you've got to have very clearly defined endpoints. Next. And all trials are registered with the clinical trials either in the US when they're multi-centre, or in Australia. I think I show on the next slide that there is also an Australian body that regulates it, called the ANZ CTR, Clinical Trials Registry. Next. And so you can see those are freely available. So if you put any clinical trial in and clinicaltrials.gov, you'll come up with it. And so I just, Amylyx is one clinical trial that's recently been unsuccessful. FDA approved them to look harder. There was a fair, one of the pharmaceutical companies was adopting it. And so it's had a negative study that's probably meant that it won't be subject to further study. One clinical trial that we've been involved with, BLZ 945, is also not advanced because of potential side effects. SPG 302, and I can answer more about that. We've started that study at the Royal Brisbane and Women's Hospital. It's also going on at Adelaide, and there are other sites in Australia. It really is not a proper phase 2-3 study of efficacy at this point. Lighthouse 2 is. So Lighthouse 2 is a study of an HIV or antiretroviral therapy, and there was an initial earlier phase study that informed a possible benefit. So Julian Gold is doing a really good job of running a proper multinational study called the Lighthouse 2. Cardinal's using a drug which I've listed there, is currently underway. And it's no longer recruiting, but it will be assessing efficacy. Similarly, the REAL study is currently underway of that drug, and again, assessing efficacy. A study that's about to start, I think it started in Sydney, is the MAGNET study. It's about roughly 1 in 5 people have a mutation that might determine whether their disease course is slower or faster, and it's then combining it with a drug to see a benefit. The ATLAS study and other studies of the drug tofersen through the company Biogen are ongoing, and I think it's mainly Macquarie. But for people with a SOD1 mutation, Biogen has been providing compassionate access of tofersen. And then there are many clinical trials of a phase 1 nature. We've had 3 at the Royal Brisbane and Women's Hospital and Wesley hospitals in Brisbane in the last year and a half. And you could read about Monopantel, which I think is more going to be Calvary Hospital in Victoria. So I can answer more questions, but I'm keen to give Lucas a talk as well. So do we want to stop there, Gethin, and hand over to Lucas? Or I might point out that the MND Australia there has got a very good webpage. And also I just point out ALS Untangled. So if you've got a therapy that you think, I want to know whether it's likely to help, and I'm hearing a lot about it, and obviously cannabis might be an obvious one. You could look it up, and reputable neurologists, usually from the States, will write a very detailed review of it. So I might stop there and allow Lucas to just talk a bit about the regulatory authority, and I'll be more than happy to, be answering more specific questions.
Thanks, Rob. I might just steal the screen. I've just got a deck here. Have we all got that? Can we see that?
Yep, that's all good. Good, Lucas. You're good to go.
Great. So, yeah, look, my role here is just to really discuss how the medicine gets from the trials to the patient. Just a few quick disclaimers. I'm an employee of Biogen. I won't discuss any specific therapies that may or may not be currently being commercialised. And Biogen is a member of Medicines Australia, so we're under a strict code in terms of what we communicate to people living with diseases and the general public. Yeah, just quickly, a bit about me and my background. So I'm a pharmacist by background, and then added some additional study in terms of health economics to get into the area of market access, which essentially picks up on all the things that Rob was saying in terms of the clinical trials and figures out how we get them to patients in a broader sense, and broader access on things like the PBS. So, I guess this is where we start. So you can sort of see after we've done phase 1, 2, 3 trials, it's clocks up about 14 years. So it's quite a long time before sponsors look to start engaging with the health system Australia in terms of that broad reimbursed access. And just, I thought a good way of trying to explain this is, I guess, a 3-step process in getting from a clinical trial that's been positive and how it actually gets to patients. So, and I've tried to use probably the most simple example that people might understand, which is the PBS, so the Pharmaceutical Benefits Scheme. So the first step is registration. So that's regulated by the Therapeutic Goods Administration. The next step is health technology evaluation. So that constitutes an independent review and a decision by an independent committee. So Pharmaceutical Benefits Advisory Committee or the Medicines MSAC Committee. And so MSAC look at things like tests, whereas the PBAC or Pharmaceutical Benefits Advisory Committee look at medicines. And then the third stage is essentially listing. So once a decision has been made to recommend a particular treatment or a test to fund, it constitutes the sponsor working with the Department of Health on actually listing the product. So conditions of supply, contracting, and things like that. One thing to sort of keep note of is there's overlap in these processes. So they're parallel. So when you look to get a medicine through, you're often got an application with the TGA as well as an application to list on the PBS. So they may not start at the same time, but they run at the same time. So yeah, how long does it take? It depends, I guess. So the first step, if we look at the regulatory side of things, what does the TGA look at? So it looks to answer questions around: is the treatment safe? Is it effective? Where is it made? Can we trust the quality? And then depending on the different pathway that the drug might take, it can take a potentially different length of time. If it's a standard review, that's around 12 months. A provisional review, that is a special type of review whereby the disease is a life-threatening or a serious debilitating condition, and there may be, I guess, not the full outcome. It might be a surrogate outcome that or an intermediate outcome that is being tested, and the company might need to follow up with new data over a period of time. So that's a provisional process. Priority, that's another one where it's potentially a shorter evaluation time. Again, looking at things or conditions that are life-threatening or debilitating. And if it's a major advance, you can potentially save some time in the evaluation process. And the most recent one is the comparable overseas regulators. So that's still a 12-month process, but what it does is it leverages other countries that are similar to Australia and uses potentially their regulatory packages to speed up the evaluation process. Maybe not in the time, but in terms of the back and forth between the TGA and the sponsor. Now, some of you might have heard of the term orphan or an orphan designation. Now, I think I've got the criteria there, but it's important to sort of keep note that the evidence requirements for an orphan designations are still the same. So you still need to go and present the same amount of evidence. I guess the benefit of having an orphan designation for a medicine can help identify, I guess, medicines and disease areas where there's really high unmet needs or where there's quite a small or limited patient population. And in identifying that, I guess, clinical unmet need is a really important factor when things go through the decision-making committees. Second step is the HTA evaluations or health technology evaluation. And I guess to try and simplify this, there's a whole bunch of questions that need to be answered to determine, essentially, should the government fund this? Because basically when it arrives on the PBS or something's funded through the medical services or funded through MBS, it's essentially taxpayers' money. So we want to make sure that there's value for money when we make these decisions. So things that get answered within this process are what's the clinical need, what current treatments are available for the condition, how much better is this new medicine compared to what we already have, is it value for money based on the price, how many people might take this medication, what's the overall cost, and particular things that might make this process more complex and potentially longer are maybe an orphan designation. If there's a diagnostic test whereby you're identifying a small group of the bigger group, if the evidence is not, I guess if it's premature, if there aren't any randomized controlled trials or there's no randomization at all, that might create uncertainty in the benefit that the drug might give when it actually gets into the real world. And any, I guess, application that requires an economic model, which determines, I guess, the value of the medicine, those things can take a bit longer. And then step 3, which is, I guess, the listing process. So that's where there's a particular recommendation for a medicine and the company will work with the Department of Health, so Australian Government Department of Health, to determine the actual criteria, routes of access in terms of where will it be available, will it be via a hospital, will it be via a community, the timing, when will it be listed, all those types of things. So that's sort of the 3 stages in terms of getting to the end of the process. But I think what's also important to try and touch on is how can people get involved in this process? And when I say people, it's people with the disease, family members, carers. Like, how can you be involved in this process? So this has evolved quite a lot in recent years. So what I've provided here is some snapshots from the Patient Voice Initiative. Now, they're an organisation that supports patients and advocacy groups to make their voices heard with respect to getting access to medicines. So, generally speaking, when you look on the 3 sort of opportunities to get involved, there's the consumer comments, there's consumer hearings, and there's also stakeholder meetings. Now, generally speaking, the consumer comments process is sort of it's evolved from a paper-based submission process. It's an online portal now, but it allows patients, doctors, families to, I guess, submit something to or be involved in that PBAC process. And this occurs after a sponsor has submitted something, but before the decision body has made a decision. So it's sort of giving that view before the final decision is made. And that's sort of been available for quite some time. The next one is a consumer hearing, and that's generally between a patient group and the PBAC representatives. That's not available for everything, so that's upon invitation. And the next one, which is probably bigger again, is a stakeholder meeting where you have the patient group, the sponsor, PBAC representatives. And again, this one's by invitation, but really the reason why it's invitation is it's there to really only to I guess talk about and discuss unresolved issues with the application. Not in every case. I mean, sometimes these will happen before something goes in, but often we see it occurs after there's I guess an issue or a slowdown in the process. Now, the Patient Voice Initiative also put out a tip sheet, which will put a snapshot down the bottom to help patients, families, and direct them as to what these committees want to hear from people, because it's just sort of putting out we want this drug doesn't really help these people make decisions. They want to know a bit more about it. So up on the right is things that I guess they ask people to try and narrow in on, which is what is your current unmet need, and what matters most to you. And that the best way to explain that is when we have those trials and we have that one measurement the trial is based on, that might be I guess a measure that's composed of a whole different bunch of tests, and it might not be your disease or what you're experiencing might not necessarily be picked up by that one measure. So explaining to decision makers on what it means to be able to do this in my daily life, it matters, and it's informative, and those type of things may not be picked up in the measure that's in the trial. So what matters to you most, and how things, what would make a difference to you, that they're really important to hear. And if you're on treatment, like what are the benefits that you've experienced? If you're on treatment, what are the side effects you've experienced? Any consequences of any starting or stopping rules that related to treatments that you're on, and commenting on, I guess, the comparator, i.e., what is currently being used at the moment. But what's next in terms of consumer engagement? I did say, look, it is changing, and this is sort of evidence of that, even in the last few years. So some of you may have heard, maybe not. In 2021, there was a Zimmerman review, the House of Representatives review, and it looked at the process of gaining access to medicines and treatments in Australia. And recommendation 28 was specifically centered around integration of the patient voice earlier in HTA. And stemming from that and related to that is what's currently happening at the moment, which is the Health Technology Assessment Review. And that stemmed out of an agreement with the medicines industry and the government to look at ways on how we can do that HTA process better. Now, earlier this year, there was an options paper that got published, and they specifically called out areas where they can make improvement in terms of getting people and patients into this process. So a couple of things that they called out here are plain language summaries of what's in a PBAC application, engagement of consumers earlier in that process, how we can get consumers involved in the process of using more real-world data in these decisions, and potentially piloting real-time interactions with these decision makers before the decisions are being made. So this is, I think, a real great step forward in terms of getting people involved, because we do know that a clinical trial is in its own setting, and then when medicines get to the real world, there's things that we find out, and potentially find out different ways in that they might benefit. So And we really only understand that by talking to patients and people around the disease. And to, I guess, quickly just sum up, so as you can see, it can take a long time to get from the clinic through the TGA onto the PBS. You know, it's quite a long time. In terms of how Australia fares in that last bit, which is integrating with the health system and getting reimbursed, we are lagging behind there. You know, I think our commitment, and any sponsor will probably tell you this, is, you know, to work as fast as you can through this process. You know, an average of 400 odd days between when it gets past the regulatory approval and being available for people is just, it's not acceptable. It's too long, and especially in areas where there's really high unmet need. So I think, you know, the sort of message when a medicine goes into this process is, you know, to make this faster, to speed this up. It's all about all the stakeholders moving in the same direction. So, you know, sponsors, patients, community, and clinicians really have their part to play when it comes to, you know, starting that access process and taking it through the end. And just finally, yeah, just like to thank you for the opportunity to talk about access. I think, you know, I'm really passionate about it, and hopefully people have learned a thing or two about how to get access to medicines in Australia. So I'm at here. I might pass it back to Gethin.
Great. Thank you, Lucas, and thank you, Rob. That was a fantastic overview of the fairly labyrinth processes involved in trials, and then taking the drug from trial to actually into the clinic to the bedside. And thank you also, Lucas, for highlighting the HTA review. We've been making submissions to that, and it's really refreshing to see the really strong focus on better consumer engagement and accelerating the processes. And I think they heard that message loud and clear very early in the process. Certainly I was involved in a few consultations, and everyone just agreed you need to have better input into, and not just the registration process, even the initial approval by the TGA, which at the moment I don't think there's any consumer engagement option at all in there. So hopefully in the future that will be the next step, is to bring in some consumer engagement into the actual decision of whether the drug's good or not, let alone whether the government are going to pay for it. Anyway, so we've got quite a few questions here, which is great. So the first question is, how long is each phase? This is talking about the trials, I think. Is that how long is a piece of string? But anyway, Rob, I'll throw that to you.
Yeah, I mean, the phase 3 studies should be, you know, 12 months, 2 years. Phase 1 studies are often initial period of only a few months. And studies also have different periods where patients might be randomized to a placebo, but then quite commonly after 6 months, patients will all be on the active drug. That's a fairly common scenario for studies. The other thing it's worth pointing out is that studies usually have an open label extension if they're a drug that's able to be produced as a tablet form. Often if it's more of what's called a biologic drug, which are quite difficult to make, they may not be an open label extension, but most of the ones I mentioned will have an open label extension. So everyone stays on the study drug after the trials finish if they wish.
Yeah, and just going to that question, we had a question as well about the Rescue ALS trial, which was the phase 2, I think, from Clean, and that's been going on for, I think, 4 years now. So I think some patients have...
Yeah, that'd be a nice example of an open label extension. So Rescue ALS has been looking at the gold product. Phase 2 study could have made my list. You know, being run, it was run out of, I think, Principal Steve and Matthew. So they've both moved sites in Sydney. They were Brain and Mind and Westmead, but it was maybe about 50 patients, and it was designed to then inform a larger study. So there is talk of of getting funding for a much larger phase 3 study of that compound.
That's right, because I think they had a second phase 2 trial that was on part of the Healy platform trial. And yeah, they're into the phase 3. And just going on from that, sorry, Rob, last question on trials for a little bit. Could you just, we have a question, could you just give a quick overview of what a platform trial is?
Well, the Healy platform would be a nice example where you have just a single placebo group, but then you're studying multiple drugs. And so that's got the advantage that you're able to study a number of different drugs compared to a placebo. The way studies also often used to do it would be to compare with historical control groups. But in that case, most drugs look pretty good because, I don't know if patients are more motivated, whether respiratory care, other care is a little more better than what it used to be. But historical controls have sort of no longer been used. But there was a big European database called the PROACT database that was often used in comparison. So one placebo group, but comparing multiple drugs has a big advantage.
Thank you, Rob. It's a question for Lucas. Are you aware of, I guess you can't talk about the Biogen drugs, but of treatments that have been questioned for cost or been knocked back by the PBS because they feel they are too expensive?
Yeah, look, I think cost is a component of every submission that goes to a reimbursement committee. There's the cost of the drug, there's the cost of the treatments in the background. There's potentially the cost saved in I guess, translating that benefit. So it might be if the drug provides a certain benefit, there's less events that occur and there's cost savings. So it is part of every submission. I can't comment on drug prices or anything like that, but it is part of the submission. And when it's a part of a cost effectiveness submission, so when you're trying to say that this intervention is better than another intervention, the way you determine value is what's the additional cost versus how much change and benefit you're going to get. So it's definitely part of the submissions and definitely part of the process. But I think you can read, and this is all in the public domain, the things called public summary documents, and they provide a summary of the submissions themselves and what actually happened during the process and the reasons why medicine submissions have passed through or whether they failed. Sometimes they mention cost. Sometimes it's something completely different.
Yeah, and I think especially in diseases like MND, where perhaps some of the traditional quality of life metrics don't really work as well, I think it's really important to really boost that consumer input into those decision making. Like 3 months or 6 months in MND is a very different to, say, in a longer term disease, I think, and sometimes I think that might be missed by some of the calculations that the PBAC use. Hopefully, with better engagement, we can get those messages across. You also mentioned about the provisional review process, and there's the sort of, you can go priority or different assessments of drugs. Does sort of a disease like MND, which has such a high unmet need and is very fast progressing, do they kind of almost automatically qualify for those sort of assessments, or do you still have to really push the case for them to get that priority review?
Yeah, well, it's I guess when you look at the criteria, it's not just about the disease. it's about what else and what's happened in the disease, what other trials have been done in the disease. If there's other treatments, generally speaking, it doesn't get approved. It's generally there for something where there is no treatment or the disease has a poor prognosis. But as part of the provisional pathway, it's really looking at a surrogate or an endpoint it's like looking at the evidence before it's properly filled out. So if you look at probably the number of drugs that have gone through that process, a lot of them have been oncology drugs, whereby the outcome might be a progression-free survival rather than an overall survival. So we haven't, our measurement isn't the full measurement, and then it might go down a provisional pathway. And as I guess a proviso is that you might get approved with a secondary endpoint or an early outcome with the provision of you need to supply data, additional data, whether that be an additional trial showing the full outcome after a period of time. So it's not as simple as just going, this disease is bad, has a poor prognosis, we'll get a provisional, we'll go down the provisional pathway. It's got to really tick the boxes in terms of, is there an early outcome or a surrogate outcome that is really indicative of further benefit? And we don't want to wait, and it's fairly validated, then we'll go down that pathway. But it's probably also important to say that just because a medicine goes down a provisional pathway and gets recommended through the regulatory environment doesn't mean it will get it'll get through the reimbursement environment, because TGA look at different things and look at, is it safe, is it effective? That might be the case based on this earlier outcome, but the PBAC are looking at value. So just because it goes through the provisional pathway, it doesn't mean it's going to go through the reimbursement pathway in an easier way.
Okay, thank you, Lucas. Just a quick question to Rob. You mentioned that in some trials, people with FTD symptoms are excluded. Is there a reason for that, or is that common, or maybe not very often?
Yeah. So look, the big ones obviously being the C9 RF72 clinical trials. WVE-002 was unsuccessful. I think our site in Macquarie were involved, and it wasn't successful, so it didn't continue. But that actually was looking at cognitive involvement. Most phase 1 studies won't specify cognitive being an exclusion criteria, and not all phase 3 studies exclude either. So there are trials that's not an exclusion criteria, but there has been a couple of recent ones where it was that case. But there's certainly ones around that aren't.
Thank you, Rob. And we had another question then about, are there any trials for asymptomatic carriers of C9orf?
Yeah, so there are a couple of companies, WVE, I think Arrowhead, that have been looking at these gene therapy trials. I think Macquarie may have had a study that was involving asymptomatic carriers. So there has been studies, but there isn't too many looking at asymptomatic, and clearly that would be a very, very wise group to look at. But again, you need good biomarkers.
Yeah, and I think at the moment they're still trying to work out the biology of the C9orf, even for the genetic. It's not as straightforward as perhaps with other treatments such as FUS or SOD1. I think it's not as simple as just knocking the gene down or raising the levels. I think they're finding it's way more complex. So it's, unfortunately for C9orf, it's not as simple as some of the other genetic therapies. Question here about obviously it's MND is quite a complex disease. So are there and trials always seem to just address one sort of treatment, which is usually targeting perhaps only one of the symptoms or one of the causes. Are there options to broaden out into multi-treatment trials or widen the scope of some of these drugs?
Yeah, well, that's a good question. I mean, you look at HIV, where individual drugs didn't show a benefit, and then when they used combination therapies, it's now a treatment which people live with. So it's likely that therapies targeting different pathways are going to be where the true breakthrough happens. At the moment, patients can be on riluzole, not on riluzole. Patients who are on riluzole do still show a modest survival benefit, but most pathways don't overlap. But I think the future, and you know, I'm hearing of it in the planning phase, that people say looking at different mechanisms of oxidative stress might be one pathway to combine. You know, neuroinflammation's another pathway. The big hot topic is a thing called proteostasis. So the people getting protein aggregates. You know, if you combine different pathways, that's really, you know, you're likely to be making you might well be where the breakthrough lies.
So this is a question perhaps for both Rob and Lucas, coming from maybe different perspectives. Is the use of real world data to influence drug approvals, obviously. For people who aren't aware, so real world data is basically data you collect not in the clinical trial. It's collected from people just taking drugs, treatments, and you basically collect the data not quite unofficially, but it's not in that structured environment clinical trial. And there's been a big push to include this more, I think, in considerations for drug approvals and probably reimbursement processes. So I'm just thinking if you could both give your opinion, as one as a clinician and one as from the industry perspective.
Yeah, I'll start, Lucas. Look, the problem is the variability and the long survivors. You know, so people who have mainly upper motor neuron involvement or mainly lower motor neuron involvement, so flail limb or PLS in reverse, will have a long survival, which might well be 10 years, might well be more. So in that case, you know, they may have gone on to a therapy and said, well, it's really made a big difference. But it's that disease heterogeneity that makes it hard to try. You know, you've got to be a little bit cautious on too much real world data.
Yeah, I think if we sort of define, you know, real world data as observational data, it's I guess increasing its presence in the reimbursement process in HTA, and that's because we're maybe going into smaller patient groups that we've done before. It's harder to get that many patients in the trials. Maybe the diseases that the companies are looking at make it harder to do randomized controlled trials. So we are seeing more of a push to use observational data or real world evidence in probably more in the HTA rather than the regulatory environment. I still think in the regulatory environment, it's sort of lagging behind. And so there's probably a bit more use of the real world evidence in proving reimbursement or cost effectiveness rather than getting the initial approval. That's not to say it hasn't been done. I guess there's been a few number of instances in the States and in Europe where real world evidence has been used to gain the approval, and we're talking about, you know, single arms without any randomization. But as Rob said, when you do that, there's huge amounts of uncertainty. So whilst it's becoming an increasing feature, it's not without its problems. The way that we probably see it and the way that I think the decision makers see it at the moment is it's probably helpful to plug a few gaps, but in terms of telling the full story of what happens in the medicine, it's a bit difficult to do that. So I think where it's probably got its place is in supporting what's been already done in clinical trials.
Great, thank you. A couple of questions, we'll wrap it up then. So the question here about the ALS-FRS is quite a subjective measurement. Is there anything better out there, or how do we deal better with ALS-FRS?
Yeah, I mean, Andrea is quite right that it's subjective, so it has its limitations, and I mean, you've got to look. No trial looks at it alone. You're going to be looking at lung function. You're going to be looking at some other measures, whether it's neurofilaments, whether it's muscle strength testing. You're going to, the limitations of the ALS-FRS are there, but people have also gone back and looked at the studies, and sure, there's a bit of noise from the variability, but it does still inform quite well with a large enough study population. So I guess, that's an important point, that, whenever you do a study, you've got to do what's called a power analysis to say, well, can I prove a difference? And therefore, that's how the ALS-FRS has both its limitations and also its place, because it means you need to have a certain number of people in the trial to be able to show a difference because of the issues with the measure that you're studying.
Does the length of trial also help to sort of even out some of that noise, moving from 6 months to, say, 12 months clinical trial outcomes?
Yeah, so you're more likely to show a benefit with a longer study time frame, and obviously everyone wants an earlier study, a quicker study. So that comes into your number of participants in a study will be how long the study is occurring. So each study has to have a very careful power analysis. It's probably also worth pointing out two things, that if a study is not showing benefit now, they often do interim analyses throughout, and they'll specify before they start the study and say, we'll do an interim analysis at 6 months, and if there's absolutely no benefit, which is called futility, the study stops. And so there's been a few recent studies where that's occurred. The other thing when people sign up for a study that they should ask is, well, what happens at the end of the study? And it's this issue of open label extension. So those are, 2 points about the study. If you think the drug's been working, well, you probably want to stay on it.
And sort of somewhat akin to that is, do trial participants ever receive feedback during the trial as well as after it?
Well, it's a general principle that that you don't receive feedback about which group you're in the randomized phase, and then everyone would know that they're on the active drug. But when you sign up for a study, the ones I've been involved with, when the study finishes, the last patient, last visit, the sponsor, which is a company, will inform the principal investigator about which patients are on which study arm, and then the principal investigator, has a responsibility to inform the patients if they wish to know.
Thank you. And last quick question. This is a question. So are techniques like CRISPR, which is a very new technique of a form of genetic manipulation, gene therapy, subject to the same sort of processes as developing new drugs?
That's probably more one for you, Lucas. So CRISPR technology is being used for these new gene therapies, the C9orf72 and SOD1 technology.
Yeah, I thought it might be for you, Rob, but no, I think if I take the question correctly, in terms of the process of HTA or health technology assessment, the process is somewhat agnostic as to how the intervention works. So the process is trying to work out whatever intervention it is, whether it's CRISPR, gene therapy, a small molecule or a service, how is that better than what we currently have? And if it is, by how much? So whatever type of technology is, it goes through the same sort of procedure of answering all those questions. It might go to a slightly different committee. I mentioned before, you've got the Pharmaceutical Benefits Advisory Scheme and the Medical Services Advisory Committee. And if it's related to, we've seen things go, gene therapies particular or cell therapies go through MSAC, and we've seen other drugs go through PBAC, and then we've seen gene therapies go through PBAC. So it might change the different decision-making committee, but the same components are evaluated, and that is how much better is this new thing compared to what we currently do.
Great. Thank you both. And there was a massively wide range of questions there covered, and really appreciate your insights into that. So for everyone watching tonight, you'll all receive a link to the recording. Feel free to distribute that across your network so more people can appreciate these fantastic insights into how we're going to get new treatments to patients. And to access this and also previous editions, it's on our website on our research page. Look under the news and update section. And just a quick plug for the M&D Research Symposium happening at the end of August in Melbourne. So we're hoping to have some brilliant researchers for the M&D Connect session. So that'll be free for anyone with lived experience to attend that meeting. I think that's all for tonight. So thank you again to Rob and Lucas. Appreciate everyone dialing in as well. I hope you all found the session beneficial, and I hope you learned as much as I did. So, and good night to everyone.